Yeah.
Oops, wait.
Oh, okay.
Did I do it?
Did you?
I didn't put in the key, but I think my YouTube has the key from last time.
Oh,
Yeah. Okay.
so are we just live now?
I think so.
Oh man.
Okay. Hey, everybody. We are. All right, I gotta stop looking at YouTube because it's got a delay. Get out of here. Uh, we are. Here we are with. This is Kyle and Friends episode 4 with Alyssa Luck. And, well, before we even get into the topic, Alyssa, would you like to introduce yourself?
Sure. So, yeah, I guess I'll start with how I know Kyle.
Mm-hmm.
I met him at the Ancestral Health Symposium in 2017 in Seattle.
Mm-hmm.
He was giving a talk there. I believe that was when you did the talk that was an Oxford-style debate with yourself,
That's right.
which it's online, so you guys can go watch that if you want to. It was good. I was there. Um,
I am undefeated in those types of debates, actually.
yeah, it's amazing. Really incredible track record.
Yeah.
So, yeah, so we met at the Ancestral Health Symposium, which I feel like he's probably mentioned that thing before, but it's like a symposium where people talk about like nutrition and stuff from an evolutionary perspective. Um, but the reason I initially got into all of that was I got super interested in nutrition when I was a teenager because I was diagnosed with something called ulcerative colitis when I was 14. Um, so that's a chronic autoimmune digestive disease. And I tried all kinds of different crazy diets to try to deal with that. And then,
Including.
including, oh man, um, well, the first one I did was a raw vegan diet, which was the worst one. I ended up in the hospital after that one. Would not recommend. Um,
I think you just didn't detox enough.
oh my God, don't even get me started. So the book that I learned about this diet from, I ended up going back and leaving a 1-star review of it on Amazon. Um,
You
and
got him.
people replied to my review telling me that I just didn't detox enough. So, and I
It's
included
real.
in my review, like, yeah, like I even paid for consultations with the guy who wrote the book, and he just told me to drink more juices, and then I was like having seizures and stuff, and they were like, oh, you just didn't detox enough. I was like, cool.
Yeah, seizure is actually a really um, effective physiological avenue for detoxification. A lot of
Yeah,
people don't know that.
right. Maybe I just didn't have enough. I shouldn't have gone to the emergency room. I should have just let it play out.
Yeah, when they give you drugs to suppress the seizure, that entrenches the toxins that your body was trying to get out. And
Right.
yeah.
Man, those bastards in the ER.
So did you do one type of raw vegan, or did, because when I did raw vegan, I started with like the I don't know anything other than I just read a book saying cooked food is poison. Like
Mm.
literally the book was the David Wolf book. The end of every chapter has the sentence "cooked food is poison." Every chapter,
So
um,
dramatic.
and so I didn't know about like calories or anything. So I was just like, I just stopped eating everything I'd ever eaten before and just ate random, like raw green beans, like just crazy, ridiculous stuff. And then when I started to learn things, I bounced back and forth a few times between the more fruitarian type thing or like 80-20 thing with like 80% fruit and 20% like greens and maybe nuts and seeds. And then
Mm.
the higher fat type thing like avocados, nuts and seeds and stuff. And I bounced back and forth between those a few times.
Okay. Yeah, so he had kind of a specific outline targeted for inflammatory bowel disease in this book. And it was mostly, he really emphasized actually a lot of the kind of tropical fruits that Ray Pete seems to like papayas and bananas and stuff like that.
Ray Pete does not like
Then,
bananas.
oh, right, because of the thing that you just told me about.
Stress-induced chitinases. Yes.
Yeah. Well, I mean, does he like bananas that aren't stressed? Or
Um,
are all bananas stressed?
I don't know.
Or is it that you can't know?
It's just that's the only thing I've ever heard him say about bananas. I also think he says if they're not fully ripe, the starch, isn't good or whatever. And
Right.
I think he said something about serotonin.
Yeah.
Basically, I can't say I know everything he's ever said about bananas, but I can say everything I've ever heard him say about bananas has been negative.
Okay. Well, scratch that part then. But I ate a lot of bananas and dates,
That
like
was your
dried
secret.
dates. um, and then I remember my parents like trying to make me raw almond butter in the Blendtec blender, which I have like very specific memories of that. It doesn't work well. Um, and then he wasn't big on vegetables. I think I was allowed to have lightly steamed carrots and celery. So I would eat that, uh, and a lot of juices, like beet juice, celery juice, carrot juice, all that stuff. He was big on the juice.
But not green juices or
Um,
also.
um, there were probably some greens thrown in there. I feel like it's been a while, so I can't remember exactly. And also, it really only lasted like, gosh, maybe a month. Maybe it was more than a month, but it was like 3 months tops. And I got down to like 80 pounds, which I was 14, so I was the height that I am now. And right now I'm 135 pounds, and I'm like not like I could probably lose 10 pounds and be good, but like 80 was far too skinny. Um, so yeah, I didn't last very long on that diet. Um, but then I did the specific carbohydrate diet for a while, um, and that was the one that I mostly stuck with. Um, and then after I ended up having to have the surgery to have my colon taken out because the diet thing didn't really fix things. But after that, I chilled out on the diets a little bit, and just, I mean, I was low carb for a really long time, like low carb paleo, but not in a, I mean, the SCD was low carb as well because you're not supposed to eat like grains or anything like that, or really starches, basically. It's like a no starch diet. So, yeah, anyway, that's a brief summary of the diets that I did.
So not carnivore.
I didn't do carnivore. Carnivore had not gotten trendy yet when I was still in the throes of nutritional exploration. And by the time it crossed my radar, I was kind of over doing extreme diets. So, like, I've been intrigued by it, but I don't
It
really
was
have
still
any
in. It
interest
was
in it.
still a germinating seed inside of the dying, like moribund corpse of paleo. People, there were like paleo was like this vessel that it was sort of propelling other things, and it was like it was almost like a dead thing that things were like consuming from the inside out, and like having these internecine wars. Like you'd go on a paleo forum and people would be like, you'd say, I don't feel good, and they'd be like, stop eating carbs. And everything was about carbs, but then like the that was like the esoteric stuff, but then the exoteric stuff was like, oh, it's just like it's like we don't eat processed food. It was this really like dishonest
Yeah,
situation. But yeah,
yeah,
people hadn't fully just been like, okay, my brand requires me to like kind of explicitly say carbs are bad yet. That hadn't happened, I guess.
Yeah, I don't even know where I was. Like my early introduction to paleo after the specific carbohydrate diet was Mark Sisson's, Mark's Daily Apple. Like he was my dude. I read his website every day. Um, and I think then I sort of migrated to people like Chris Kresser and then the sort of Ancestral Health Symposium crowd. Um, but yeah, by then I had kind of lost momentum on the nutrition thing, um, which is funny because like I ended up studying nutrition in college because I guess just the timing worked out to where right around the time I was having to decide what my major was going to be was like the peak of my interest in it. Um, so I don't do anything with that now professionally. I just have a nutrition degree.
Isn't that
But
funny? Like it's basically I think the lesson here there's a quote floating around. I think it might be new from
a new quote.
Ray Pete
Oh, oh, okay, of
that
course.
is something
No other
like
quotes matter.
yeah it's like if you find yourself focusing less on I forget whether he said like your health problems or your like diet stuff or like just like that's a good sign.
Mm,
Um, and that kind of speaks to that. It's like, oh yeah, this is so important to me that it's going to be like my college major. But really,
yeah,
that's just kind of like a side effect of like that you're not like in a good place with your personal health. And not to say that
right.
everybody that studies nutrition, it's kind of this weird thing. It's like I think there's an old joke like psychologists all are mentally ill people. um,
Yeah,
it's,
yeah. Well, I mean, I wanted to do something with it. I was like, oh, I'm gonna like figure out how to cure myself, and then I'm gonna help cure other people, so I can go to medical school, or I'll go, I don't know, become a chiropractor or an acupuncturist, or one of these people who can like see patients and help them. And that definitely didn't end up happening. Would not have been the right move for me. But at the time, I was super into it and really wanted to do that.
yeah, yeah, I definitely went through a few incarnations of like, oh, I want to do this, I want to do that. But really, I think I was talking to myself. You know, I want to help. It's like, oh, I want to help. And it's like, I really wanted to help myself. And okay, so you mentioned Chris Kresser, for whom you worked.
Yes,
You
for a period of time.
so there's a like a how many blog posts do you think you wrote for his website?
Um, I don't know. It was let's say I was on his staff that helped research blog posts and
Hmm.
helped put some of those together. So it's kind of hard to say how many I had a hand in or helped with.
Right. So it might just be interesting to people to know because I didn't know this until I guess I could have guessed it, but um, so I don't. I've never. I don't know how to say this. Not that I like produce a lot of content or anything, but I've never really like had somebody else do work for me,
Mm,
like on anything. Um, and part of that is that I don't monetize anything, so I can't pay anybody to do it. Part of that, I think the biggest part is I just have obsessive compulsive disorder, and it would probably throw me for a really big loop to give over almost anything to anybody else. That
yeah, yeah,
so like
yeah.
for a number of reasons.
I'm remembering you watching me putting away silverware
Mm.
and getting visibly anxious because I wasn't doing it right.
Yeah. Yeah, you know, it's like but yeah, it's kind of like I just have to not like look at that stuff.
Mhm.
So yeah, but apparently, you know, people that produce a lot of content um have like staff and it's not really advertised or put out in front that this is like a composite product or something. But
Yeah, Although
basically.
I was looking at his website, like just a couple of days ago actually, and he has a page there now, like introducing his whole like research and writing team. Um, it's pretty big now. His company's grown a lot. And also, I recently, I was just checking Denise Minger's website because, as you know, I'm obsessed with her. Um, and she has on her about page that that's the kind of work that she does or has been doing, is like helping these big gurus like Sarah Ballantyne, the Paleo Mom, and people like that with like research and stuff for their programs.
Well, I don't like that. Um, I,
Well, I don't like it either because I think that Denise should be writing her own stuff under her own name because she's the best and I love her. But maybe that's neither here nor there.
yeah, that's actually kind of a weird move for somebody like her because she has a ton of name recognition, so she doesn't really
Yeah,
need, you know,
right.
that's weird. I guess obviously
I
it's
mean,
her choice though for some other reason.
well, I'm sure there's something to be said for like having somebody pay you versus like being responsible for your own like monetizing your own content somehow, you know?
Yeah, 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 for sure. Um, okay, so we've got somebody asked, "Are we dabbing on polysalids today?" No. Uh, Also, there was a bit of an echo
That ain't me.
apparently that was fixed, but if you go back and watch
Oh,
this, there's probably you probably might be a little disappointed to know that at least half of your introduction had an echo, but it's not like it was not.
oh,
I didn't hear it, but
where is an
oh
echo?
no,
Is it gone
it
now?
was me. Well, it
Oh,
was OBS. I just forgot to turn off the some of my like the default when you just open the program. It has desktop audio, so like your audio was coming through my audio input and then being sent out, and then also your
yeah.
audio was being sent out as an NDI source.
Mm,
So I just had to X those out. Um,
okay. Well, I just won't ever be able to listen to that. That's fine.
well, I'm kind of curious how bad it is, so I'm definitely gonna. Well, somebody said, they were like, you have a bit of an echo. Yeah, sound is weird. Uh,
Mm.
Echo is even worse now. um,
Great.
Matt asks, does canola oil reverse fatty liver disease? Absolutely. Um, studies, a study, uncited, shows that canola oil did better than butter in the.
Which was also olive oil.
Yeah, yeah, yeah. When butter and canola oil were pitted against each other, olive oil won. So,
Yes, exactly. Nailed it.
um, okay. So, just a real quick correction. We actually met in 2013. This we definitely actually the one that I'm less sure about is 2016. Uh,
I oh, right, I was there in Boulder.
yeah, were you at all of the AHSs after from
Mm.
2013 on?
No, I was at 2013, 2016,
That's
2017,
weird. Okay.
and then 2018, which you weren't at.
Yeah. Yeah, I had officially been done with that.
Yeah.
Um, which there's actually something on the back burner about.
Yeah, I hear you had a really good talk planned
Yeah,
that everybody would have loved.
yeah, well, there's that, and there's also the sugar paper, sort of the review that I will finish one day, and then unleash the criticism of the AHS, the whatever that journal that I guess is defunct now.
Oh, right. Yeah.
Why they didn't want to publish it and all of those shenanigans. But yeah, I definitely we definitely met in 2013 after my first talk, which is lost to history. My first talk was more or less my thesis proposal that I would have given to my dissertation committee in 2013, and I also gave maybe a little bit more entertaining and less like strictly the way the proposals have to go in graduate school version in Atlanta. And yeah, they lost almost all those talks. And it was funny, a couple years, wasn't it a couple years ago, they found a bunch of them and published them.
Yeah.
And yeah, mine
Yeah.
wasn't one of those. So, um,
Yeah.
okay, so here we are. We got some showmanship here. We're 20 minutes in, and I think now thoughts on probiotics. We got a comment thoughts. Perfect. That's a great segue into the topic, which is going to be this sort of a sub. Uh, I like to talk about good science and bad science and fads and stuff. And some of the best examples. I mean, one that's almost just a slam dunk now is cholesterol causes heart disease. Dietary cholesterol causes heart disease, and dietary saturated fat causes heart disease. That's obviously something that was like taken as self-evident for a pretty long time, and now it's sort of almost a joke. And then the early omega-3 stuff is getting, I mean, that's still being heavily marketed, and I think it's got some juice behind it because people do have acute positive effects for reasons. But basically, all of the big trials, like showing reduction in cardiac disease, that was the early successes that they all failed to replicate later. And then the sort of newest one, which is getting a little old now, but it's still, I guess, in sort of a fad. I think people are kind of bored with it. I just don't see as much excitement online about probiotics and the gut microbiome.
Mm.
Um, but
Do you
yeah,
think that's just the circles of people that you like interact with? Because it seems like, at least, like it seems like in the mainstream, it's still
there's a peanut
very
butter
like
egg.
up. Okay. I thought it was a Cadbury egg, but then the middle looked a little off.
Oh, I have some of those too that I'm going to eat. See, the peanut
Okay.
butter egg brings you down, and the Cadbury cream egg brings you back up. So you gotta.
Yeah, like to me, it seems like the in the mainstream, probiotics are still on the rise as far as more people knowing about them, more people taking them, that kind of thing. Do you just mean like in the research science realm, or in the like die-hard health fanatic realm?
Yeah, more like the forward-looking, whether it's the research. I think this is a little hard to explain, but there's a certain energy to topics, and when you see them showing up in some places and not others, and a certain amount of and style of excitement, and the research being talked about in a certain way, it's kind of the future, and that would have been probiotics like 10 years ago, and now it's kind of like it has changed from Apple 20, 25 years ago to like Apple now. So it's still
Mm.
profitable, but you're not gonna like buy shares in like big probiotic or big microbiome, um, macrobiome, and get rich quick. Like you might have if you started making content about that. Like if you had gotten in on the ground floor of being like the person online talking about the gut
Right.
microbiome 15, even 10, even 7, I don't know, years ago, you could have made like a big career.
Oh yeah, totally.
That's pretty much over. I mean, you can still make a career. You can always make a career about anything. It's just you actually have to be really good and like compete.
Yeah.
But when things are on that growth phase, you can kind of suck and still like blow up and you know.
Yeah. Well, and I feel like now you would have to be a lot more specific, and just focus on like a niche area of microbiome research or gut health, because the content arena there is just so saturated now, and everybody's talking about gut health all the time. And there are like certain things that are now common knowledge, like, oh, bone broth and fermented foods and all this stuff for your gut.
So how about
So
we start with that? Because somebody asked about probiotics, and one of the ways that I feel that the gut stuff, its days are sort of numbered as like the fad is over, is whether it's fermented foods. I mean, fermented foods are kind of, because they're food, so they don't really
Indeed.
have to be medicine totally. Like even if whether it's like yogurt or sauerkraut or kimchi or whatever, even if they don't meaningfully change the bacteria in your gut, like which they don't seem to really do, that that doesn't really, like, people aren't just going to like throw out all their fermented foods because maybe they like them, and maybe the fermentation process produces some vitamins in those foods that you can get, whatever. But probiotics, like pills and or whatever, those are medicinal, and so if they don't meaningfully change the composition of your gut microbiome or provide some other type of therapeutic benefit, then they will be abandoned because nobody takes those for any other reason. And so I think we should just start there with big picture stuff. Like, is it true that the vast majority of uh, fermented foods, which again, this isn't as big of an indictment against them, but probiotic products don't sustainably, change. At best, some of them change your gut microbiome while you're taking them, but it'll just revert back to what it was before, like very rapidly when you stop. Um, would you agree?
Yeah, so I feel like that's probably one of the biggest like misconceptions about probiotics still in the mainstream is it's actually like in the literature they went through this paradigm shift a while ago from oh probiotics are like reseeding your gut with good bacteria um, to no they don't actually do that like these bacteria that you're taking orally have they're transient they don't really take up residence in your gut. They don't stay there. Um, but that doesn't necessarily follow that they don't have therapeutic effects. So sort of the general consensus now, I believe, is that probiotics still have therapeutic effects through things that I wouldn't be able to do justice to right now because I was not prepared for this question. But via like immune modulation and stuff like that. And it's actually prebiotics, so fiber and such, that meaningfully change the composition of the gut microbiome. Like, permanently is the word I'm looking for, but like in a sustainable way. Although, even then, your gut microbiome changes very rapidly in response to dietary changes, which is kind of related to the topic we were going to talk about today. But so, like, even then, it's not like you can take prebiotics or eat a lot of like onions and artichokes and whatever, and then stop eating those things and maintain the gut microbiome that you built with those foods. Like, it's always shifting, which makes sense because they're just adapting to their environment. Like if you're eating foods that feed a certain kind of bacteria, you'll get more of them. If you stop eating those foods, they're not going to be able to keep surviving at that abundance.
Right. So somebody asked, which I think is the one thing you didn't touch on in that broad like overview, was the spore
Mm.
products. So my
I
understanding
don't
of
know a lot
those,
about those.
yeah, my understanding of those is they're transient. They don't actually colonize. I'm not even sure the spores take up residence as like mature normal bacteria. Probably some of them do in the process of their transient relationship. But I think a lot of the effect that they have, which is an antibiotic effect on other uh, microbes, which are generally considered not good to have, is from
Yeah.
just them in their spore state, in the spore phase. Which makes, I mean, if you just think about it, so I don't think anybody's actually ever said this. I've never heard this, but just stop and think, right? You are a bacterial species. you're under a lot of, because what they do is they make these endospores in situations when things are pretty bad, and for them, like that they don't. It's like the whole just dividing and thing is this not appropriate for right now. So they make these really hardy, like super hardy. These endospores are the reason that autoclaves have to use like steam heat and a certain amount of time, because they're the ones that really require, like, regular bacteria almost completely are destroyed from like regular boiling. But to really, get that like 100% sterilization for lab work, that's because of spores or certain types of spores. And so if you think about it, what would they do to competitor? What would it be good for them to do to competitor bacteria, these spores? Like have things on the outside of them that are antibiotic to those competitive bacteria. Like that makes sense, right? So just real quick, like it's not like a magical. This is just kind of, it just kind of makes sense. It's not unlike the Ray Peat thing from a few years ago when he started talking about mushrooms and how they're antibiotic, because they digest things. They have like an outward facing digestive process as opposed to an inward facing one. And so they're competing. They're fungi and they're competing with bacteria. So yeah, they're gonna like have things about them, specific chemicals, as well as just sort of a general physiology that it tends to be antibiotic. Yeah, somebody said
Makes
there's a
sense.
study showing lower postprandial endotoxin. Yes. So that is the effect that the spores do. They
Was that study on a specific like brand that is on the market? Because I know there's like the Megaspore probiotic. Is Just Thrive, is that a spore probiotic brand, or am I thinking of a different thing?
yeah, I think so.
Because I know that's been a popular brand, but I can't remember if it was the spore one.
Yes, I think so.
It is.
And that is to
They're,
say, I
they're
think that brand has an endospore product. I don't know if it has also
okay.
other probiotics, but I'm like 99% sure. Yes.
Okay.
The,
I remember one of their marketing claims was like, this turns your gut into an antioxidant factory. And actually, can I just put a plug in here for Lucy Mailing if you guys are interested
sure.
in probiotics? She's one of my friends. I also met her at AHS, and she writes about gut health and stuff. But she has a really, oh, you know what? Maybe it must be a soil-based organism because I think this article I'm thinking of, she reviewed all of the like soil-based probiotics. But she did like a super comprehensive review of all the like semi-popular products out there and checking to see if their marketing claims actually match any research at all. And I actually started taking one of the probiotics that she ended up recommending based on that. There was only one that she thought actually lived up to the claims. And it seems to be helping, which is weird because most of the time when I try things, they don't help.
So true. I tried some of the stuff that she recommended. The butyrate. I've tried a couple brands of butyrate. The thing is, though, usually when I try things, I kind of try 2 or 3 at once. Like she had 2 or 3 at once recommendations in one of her articles, and it's really hard when you're like too OCD or too like autistic about it, because you start psyching yourself out where it's like, okay, I just need to try this one thing to see if this one thing helps. But then but what if they all potentiate each other, and oh, but what if like one of them messes up it's like,
Oh, I know how it goes.
you kind of almost have to be grug about trying things. You have to just be like, this thing do this thing good work now
Yeah.
or not. Um,
Yeah.
yeah, there's like an infinite number
Well,
of ways that things can go wrong, and
yeah. Well, and with the butyrate, like if you already have a fairly like healthy large intestine and like a have pretty like hypoxic environment there and like decent balance of gut bacteria, like butyrate in a pill isn't going to really like sway the balance if you already have like healthy butyrate production from like your native bacteria. So I don't know.
Word. So, so real quick, yeah, the spore probiotics in general, as far as I know, just any of those products probably do the thing of lowering postprandial endotoxin because they have an antibiotic effect while you're taking them on the bacteria that produce endotoxin in the small intestine, probably also in the large intestine, but the small intestine is more relevant for postprandial absorption, obviously. So,
But also you should check on the research that those companies publish and make sure it's actually like, make sure they aren't just saying that, because a lot of effects are like pretty strain specific. So like if a study's like, oh, like this strain did this thing, like you want to make sure you're taking that strain specifically. Otherwise, you don't necessarily know
right.
if you're going to get the same effect.
Like if we have this strain on their website and then their website links an article and it's talking about a different strain, that's usually a bad sign. Yeah.
Yeah. Yeah.
So that's the one thing. The other thing we mentioned, which I guess we should just clear up, is the butyrate thing, which is that butyrate from, so it's I guess it's the shortest chain fatty acid. I think anything less than.
I think propionate, isn't propionate 3?
Yeah. I didn't know if that was still called a fatty acid.
I think so. Actually, I'm not 100% sure of the terminology, but some bacteria make it and it's good for your colon cells still.
Yeah. I guess 3, 3's company too. And when you get down to 2, then you're just at an acetyl situation. So yeah. So it makes the penultimate smallest, or anyway, bacterial fermentation of fibers produce quite a bit of butyrate, propionate, other short chain fatty acids, and they are, and this has been shown actually. This is pretty solid. They're the colonic cells. They really like them. They take them up and use them for energy production. And the net effect of this is they're sucking up all this butyrate. And in order to, so it's a fatty acid, right? So for them to use it, and they use this preferentially even over glucose when it's present, they can't ferment it like glucose, so they can't do any glycolysis with this. So all the energy these colonic cells are getting is from full oxidation of these fatty acids, and so that requires oxygen, and they pull a lot of this oxygen out of the lumen of the colon itself, and this creates like a semi-hypoxic environment, which itself apparently, and I think this is pretty good science. I think this has been shown in a number of different places. Is that state of the colon itself is a very powerful controller of the amount and the type of bacteria that grow in there, like way more than, for example, the introduction of bacteria. Like if you were to take a probiotic or something, that versus if you were to disturb the oxygen content of your colon, like that would have a much bigger and longer lasting effect on the composition and the amount of bacteria. And then further up in the small intestine, and I think this isn't quite as good because the small intestine is just real funky and real, it's like the glutamine thing, like how so the small intestinal, the enterocytes there, they seem to like the glutamine and glutamate, which I suspect is to keep too much glutamate from getting into the system because it's excitatory. So they kind of suck out a certain amount. But at any rate, that's how they sort of keep the small intestine relatively hypoxic. But I don't think it's as hypoxic as the colon, or I don't think it's as big as an effect and has shown as much therapeutic potential. But at any rate, that's the butyrate connection.
Yeah. And just to tie it back in, the whole thing that you just said explains why prebiotics have an effect on the microbial composition of the gut much more so than probiotics do, because prebiotics are what feed the bacteria, which then ferment these to produce the butyrate, which then do that whole thing that you just said.
Right. Yeah, because the bacteria are there. I mean, even if you have, dysbiosis or whatever the heck you have, there's basically never a situation where you'd have like zero of the bacteria you want or whatever. So yeah, so like introduce it's not like, oh, I had an antibiotic because I had some type of medical situation for which I needed an antibiotic, killed a bunch of bacteria in my gut. It didn't recover the same way. I've had gut issues since. I just need to reintroduce what was missing. That's really just doesn't work. But what seems to work is to try to do this thing where you like mimic a healthy environment and it'll sort of naturally. And I think actually that can be sustainable. Like if you can get that to reverse with, say, taking just straight up butyrate or something, I think you don't have to take that forever, because once you get the hypoxia going and you get the bacteria back that are making butyrate, you could like get
And are eating enough things that allow them to make butyrate.
right. So this isn't for carnivores.
Right. Unless they're eating, I don't even know how to
Chitin.
say that word, animal fiber.
Yeah. I'm not sure that that's really fiber that can be made in.
Yeah, can bacteria make that into butyrate? I don't even know.
I'm sure there's some bacteria that can. I'm not sure it would meaningfully happen to a meaningful extent in the colon, but whatever. Okay, so that's some general stuff. Probiotics are mostly bullshit. To the extent that they have an effect, it's generally only when you're taking them. I think one caveat is that there have been shown some long-term effects of the fecal transplant procedure. Is that right? That like
Yeah, yeah. Well, especially with like C. diff, it's
right.
still results are still quite mixed with like inflammatory bowel disease. I actually did a fecal transplant trial for not for ulcerative colitis because at that point I didn't have my colon anymore, but it was for pouchitis, which is basically the version of ulcerative colitis you get after they've taken your colon out. So it's like
Because
in the end of your small intestine instead.
they sort of make faux colon, right?
Yeah,
Or yeah.
yeah, from your like ileum, from the end of your small intestine, and then that can get inflamed. So yeah, it like didn't really do anything for me. But yeah, the evidence is mixed there.
Yes, indeedy. We got
And I would just like, I don't agree that probiotics are bullshit. Like, they don't they don't change the microbial composition of your gut, but they can have therapeutic effects for people. But again, like while you're taking them. But if you have serious gut issues or like a disease or something, if you can get benefit from something while you're taking it, like that's still a really good thing.
yeah, I guess so. I guess I just mean like the culture around probiotics is bullshit. Like when the average person, even the average oh wait. Looks like something has frozen. Uh Are you frozen or are I frozen? Frozen. Hey.
Hey, are we back?
Yeah, I think so. Okay, I think it was
Okay,
you.
cool. How
Yeah,
do you know it was me?
because somebody said so in the chat. I mean in the comments.
Oh, sorry.
Also, your window size changed.
Oh,
I just changed it back.
and I'm
Hardwired.
plugged in via
Yeah,
Ethernet cable right now. That's how committed I am
I know.
to my internet bandwidth.
Damn it! Now I have to change the window size again. Oh, I forgot. Oh, that took 5 whole seconds.
I was gonna say, does it really take that long?
No, no, it doesn't. Um, all right, we're good now.
Okay, cool. We're back.
So, what was I saying? I was saying that when the average person and even the average like health, I like follow some accounts online. Maybe I even read a study or two once in a while. Person thinks about probiotics. They have a
Mm.
certain narrative in their head that has been
Right.
heavily marketed.
Yeah, they think about the reseeding thing.
Yes, that's
They
kind of
don't
what it
think of, yeah.
means. That's really what it means. All this other stuff again is like yeah, it's it's interesting too because a lot of the things that people talk about, like one of my committee members on my dissertation committee did research on probiotics, and she didn't even really have a perspective. She didn't really know the difference between colon versus small intestine microbiome. And I thought about this.
Well, that's almost fair because I feel like nobody talks about the small intestine bacteria except like in the context of like SIBO or something.
Yeah, that seems really dumb though. And I was thinking
I would
about
agree.
it, and I think the reason why is I worked with her a little bit, not just as a committee member, but she had mice, and that I guess they were collecting like fecal stuff from to do sequencing of the microbiome, and then I guess our lab was collecting other tissues because they didn't need the other tissues, and it was an experiment that was sort of relevant to something we could do, whatever. And then we learned how to collect the fecal matter and started doing that. I don't think I ever did any sequencing on my mice, but I did collect feces from them. And what I mean by that is the way it's done. So with rats or mice, I don't know for other model animals. Obviously, for humans, they can do like aspiration of stomach contents, small intestine contents, scopes up the other way. But these other animals I mean, mice are tiny, tiny, and even rats are, I think, too tiny for that. I think maybe you can get stomach contents from a rat, like get an aspirant. I think it would be a little dangerous to do. Like, I think it would have to be a procedure that you put the rats to sleep for, and it would be a pretty serious, like, skill based procedure to not mess up their small intestine to, like, get a thing in there, like, through two sphincters and, like, get stuff and actually. Anyway, what is done is you kill these animals and you take out their intestines and you just there's the small intestine and you kind of follow, and it's actually mostly well, see, because the further up the less solid they are, but you basically you can find throughout the tubing of the whole GI tract like a pellet forming of feces, and you basically just clip with scissors that piece out and then squeeze it. And you're supposed to not touch it because you're not supposed to like introduce anything like on your tools or whatever. But you basically have a tube that you would collect any kind of tissue sample in, and you squeeze like a sausage this forming fecal matter into a tube. And because of the nature of like those are just wherever they happen to be in the GI, from top to bottom, you just take what you can get. The further up it is, the more watery it is. It just doesn't come out. Then the further down it is, the more it comes out like a pellet, and it's nice. So just because of that practical reason, a lot of this sequencing stuff is done on these model species, and there's really not a good way to or if there is, I mean, that people don't do it because that's what I'm saying is that's what people do. And that kind of technique would not lend anybody to think, oh, what's happening to the large versus small intestine microbiome? Because people don't think of things that would require them to stop using the technique they're using that everybody in their lab knows how to do. That's like a well-oiled machine, and to do something else. That's just it's just not how it is. So anyway, so that is our overview of the gut microbiome field, and now we're going to get into shenanigans here. Specifically with, so you started, it's been, I might overshoot the time here. I'm going to say half a year. Is that too much? Okay, a quarter of a year?
Because the study was published in October of last year. So that's when I became aware of it and fell down this whole rabbit hole. So it was like end of October.
That's half a year ago, by the way.
Really?
October,
No.
November,
Yeah.
December, January, February, March. That's 6 months, which is half of a calendar year, a Gregorian calendar. So, but so you probably started this project something like 5 months ago. You started maybe thinking about it, and you started working on it probably more like in a serious way, probably more like 4-ish months ago.
Yeah. Well, I'll just
Okay.
I'll just say what happened.
Okay.
So,
but don't mess up the timeline though.
Okay.
6 months is half a year.
We have the rough timeline that I will be attempting to work within. So I get Chris Kresser's newsletter, and sometimes he just has links to studies that he deems important or interesting, or probably more often it's like studies that make some kind of popular science headline. And that was the case here. So I get his newsletter, and I see a headline for high sugar diets damage the gut. And being the, sort of repeat-minded, adjacent person that I am, I was like, I'm going to investigate this. So,
would you like to know more? Click.
Yeah. So I opened the media coverage of the study, and the title that they went with is High Sugar Diet Can Damage the Gut, Intensifying Risk for Colitis. Like, well, that sounds pretty bad. And then, they have their little summary. One of the things they said, mice-fed diets. So this is a mouse study. Mice that were fed diets high in sugar developed worse colitis, a type of inflammatory bowel disease, which already, I just wrote a whole article about this as well, like what you can actually know from mouse models of colitis. And it's really, it's not accurate to liken a model of mouse colitis with human inflammatory bowel. It's a model that people use, but it's not like a good model. But anyway, that's a digression. So
we should get back to that. Put a bookmark in that, but yeah, continue.
Okay, we'll put a pin in that.
Yeah.
High sugar diet can damage. Mice fed diets high in sugar developed worse colitis, a type of inflammatory bowel disease, and researchers examining their large intestines found more of the bacteria that can damage the gut's protective mucus layer. So at this point, I'm like, all right, what is this bacteria that can damage the mucus layer? Because that sounds pretty bad.
Mm-hmm.
So I look at this paper, and actually I sent the link to you, and you sent me the paper. So thanks.
Yes, that's Chi-Hub dot.
Yeah.
What is Sci-Hub dot? It's not .com, right? It's like dot.
Oh, I don't even know.
It's dot. Yeah, but this is
Chi-Hub. Dot info.
Yeah.
Dot biz.
Yeah, dot biz. Yeah, that's all the.
Yeah. So this paper, they, do their whole introduction, whatever. So this is the relevant point where they talk about this horrible mucus degrading bacteria. Erosion of the mucus layer in sugar-fed mice is explained by our microbiota analysis, showing that the abundance of Akkermansia muciniphila, a mucin-degrading bacterium highly abundant in the human gut, is increased upon consumption of high sugars. Now, this is the only background context that they give on this bacteria. Recent studies have documented beneficial roles of Akkermansia muciniphila in human health. They cite 2 papers. However, there is also substantial evidence that A. muciniphila promotes colitis pathogenesis. And they cite 3 papers. So I was like, okay.
Seems contradictory.
Yeah, well, it's like, I'm going to learn more about this bacteria because it sounds like, well, yeah, A, that sounds contradictory, but B, like, they're framing this as a bad thing, and they're very much, like, citing, like, positing this as the mechanism for these high sugar diets like, dramatically worsening the colitis that these mice had.
By the way, so are these mice a special strain that have a proclivity towards this type of?
They used several different models. They did a bunch of different like sub experiments. So some of them were, some of them they just used the DSS model of colitis on wild type mice, and some of them they used like I think interleukin 10 knockout mice.
Okay, and the DSS model, that's they induce that with?
Yeah, they just give DSS in their drinking water, and it causes all this. Like it causes basically massive epithelial damage to the lining of their colon, which then causes inflammation.
And DSS is?
Some chemical.
Okay. So I also forgot the acronym, so I was hoping you would cover for that, but neither of us.
It's not important.
Yeah, but yeah, I'm sure it's important. This is like an established, like this is the thing that people do. That.
Yeah, and like I mean, I guess you could link it in the description if you want. But I just put an article on my website about like all of the different mouse models that are used for colitis, and I talk about that one. And I'm sure I have the full name of DSS in there.
Yeah. Oh yeah. The description will be lit for this one because we'll have like all the papers and then your stuff, and it'll be great.
Cool. So to continue this story, I Google this bacteria, which they made sound very harmful to the gut, Akkermansia muciniphila. And I'm kind of surprised to see all of these articles talking about how to increase levels of this bacteria and how it's really good for gut health.
Totally.
And yeah, and a company selling it as a probiotic for diabetes. And people talking about how this bacteria, which again, these people said degrades the mucus in the gut to an extent where it worsens colitis.
Mm-hmm.
So, and I'm coming across all of these articles with people saying, oh, like it actually strengthens the mucus layer of the gut. And I'm just like, all right, so we've started something now. Like this investigation is happening.
Hard for both of those things to be true. That's tough.
Yeah. Right. Yeah. It's hard for it to both harm the mucus layer and improve it at the same time.
Yeah.
Although, like, I will say, like, in all fairness, it's not impossible simply, like, obviously in the same, like, moment in time and space, like, it's impossible. But, like, things can be heavily context dependent. Like, you can have something that is perfectly functional and healthy in, like, a healthy state of homeostasis, but that can have different effects in disease states. And they call bacteria like that pathobionts, I think. Right, like where they're good.
What's it called? It's opportunistic pathogens.
Yeah, something like that.
But that's not so. But to be unfair or to be harsh, that's true. But also probably almost never included in the discussion in a lot of these experiments. Like,
right.
if they're just saying this causes this,
Right. And especially once it makes it to the media, nobody is like
right. Yeah.
having that discussion with any kind of nuance. And yeah, just to clarify, pathobion is a commensal species that can become a contributor to disease in certain contexts. So like, to be fair to these researchers, like in their paper, I think they paid, actually, I'm not 100% sure about this, but I think they paid very brief lip service to the idea that, in some, like, this bacteria isn't all bad. It could be classified as a pathobiont. But anyway, so, after my initial Google, I went to actually looking at the scientific literature, not just random articles on the web, about this species of bacteria. And I, of course, started with the papers that this sugar paper cited, again, saying substantial evidence that this bacteria promotes colitis. I guess to them, 3 mouse studies is substantial evidence.
Well, it's certainly substantial if there weren't opposing studies.
Aha. Yes.
It is substance. It has,
Right. It has substance.
Yeah.
Yeah. So here's a question for you. How, like, if we're talking about like expectations for scientists to be aware of what's going on in their field, like, I'm not really familiar with how long the process is from like submitting a manuscript to it actually being published. So like, what time frame would it be like reasonable to expect that they would have seen something? Like, obviously, if something is published in like March 2020 and theirs is published in April 2020, like, I can't be like, oh, how did they not know this study existed?
It could be as
like,
much as a year.
Okay. So that's kind of what I was thinking.
Yeah.
In my like, trying to be fair.
Yeah, that would be that would be pretty fair.
All right. So I pulled some of the titles of some other studies that had been published about this bacteria. So the one I'm talking about and the sugar colitis one was published October 2020. So starting at the earliest, in 2013, we have extracellular vesicles derived from gut microbiota, especially Akkermansia muciniphila, protect the progression of DSS-induced colitis. Seems relevant.
Protect.
Seems like maybe they...
As in protect against the progression? Okay.
Yeah. That's what they meant.
Okay. Yeah.
So, that seems relevant. Like, maybe they should have known about that.
Yeah, right in the title. That's actually very good for scientists.
I'm only referencing ones here where it's in the title.
Right.
Like, I'm not even expecting them to read the abstract here.
Yes.
We're having expectations low.
So this is like, stop being confused about health level. You should have picked this up.
Yeah, I'm just going to stay out of that one. So then we have, in 2015, Akkermansia muciniphila adheres to enterocytes and strengthens the integrity of the epithelial cell layer. So that was actually an in vitro study. So what you can conclude from that is, a little bit beside the point, because it seems like they should have at least known that it existed. 2017, so we're still 3 years out here. Chlorogenic acid ameliorates experimental colitis by promoting growth of Akkermansia in mice.
Because those people probably are already assuming See, that's a good example of the progression of the literature. Is that at this point it's kind of established that that species does that effect. So you could just say, "Hey, look, this promotes that species," and everybody would go, "Oh, the one that has that effect." Like that's kind of
Right.
how, yeah, sort of like
Except
how
it's crazy.
cholesterol, like, oh, this increases cholesterol, and then everybody just assumes, oh, that's going to be bad for my heart.
Right.
Hmm.
Yeah. But like it's weird that people would do that in this context where experiments are finding totally opposite things.
It's a certain kind of weird, yeah, that we have come to expect.
Yeah, yeah. All right, so I put these out of order. So back a year, 2016,
Okay.
caffeic acid. So this is a different paper. It's by the same group. Caffeic acid ameliorates colitis in association with increased Akkermansia population in the gut microbiota of mice. Then in 2017, we have a nice little review entitled Next Generation Beneficial Microbes, the case of Akkermansia muciniphila.
Is that the only one that they list in the title?
Yeah, that's
Wow.
the full title.
Did they talk about other strain? Because it sounds like they're saying next, like, here's a list, a breakdown
Yeah,
of.
no, they're only talking about this one bacteria.
Wow. So that's got single sole billing as next generation beneficial bacteria. Okay.
Well, it's using it as an example,
Okay.
and just focusing on that. But yeah, um, and then here, okay, so you tell me what you think about the timeline of this. One of the papers that the sugar study cited as substantial evidence that this bacteria promotes colitis was done in interleukin 10 knockout mice. Another group of researchers tried to replicate that exact experiment and failed to, title being Akkermansia muciniphila does not promote short-term intestinal inflammation in gnotobiotic interleukin 10 deficient mice. So this earlier study with this negative result, like I feel like I never actually see this happen in science, but like the point of science is to like replicate experiments and like either confirm or like that's kind of traditionally how it's supposed to be, right?
That's what they say. Yeah.
That's what they say. Okay. So this is one of the few times I've seen somebody actually try to do that,
Hmm.
and they did not confirm the result. They found that that didn't happen. So that was published September 2018, which is a full 2 years
Yeah.
before this sugar paper was published.
Yeah. That's fair.
So,
Okay. So going back to the sugar paper, you're what we're getting at is they didn't cite any of those. Is that correct?
none,
Okay.
none.
And
They
those
showed,
would come up in a search just using the species
yeah,
name of that that they're studying. Okay.
yes,
Just to make
correct.
that clear.
Yes, exactly.
Yeah.
Yeah, they not only didn't cite any of these, they showed absolutely no awareness that there was like I mean, they did say that sort of contradictory thing. Recent studies have demonstrated beneficial roles. However, there is substantial evidence that it promotes colitis pathogenesis. But like they didn't show any indication that specifically in the realm of colitis
Hmm.
in mice, there is controversy. They just
Right.
they just stepped right on over that
What did
because
they
the paper.
cite in the? I mean, I don't. Maybe you don't have that written down, but when they
Yeah,
said
well, I
some
remember.
studies, okay.
They cited so they for the beneficial effects one, they cited one study. They were both related to obesity and metabolic syndrome, because that's actually where most of the research has been happening on this bacteria has been in that realm. And like I said, there's a probiotic on the market for diabetes with this strain in it. Um, so yeah, it's apparently really good for obesity, diabetes, metabolic syndrome. Um, yeah, they like it's just insane to me.
Right. Okay. So I want to go back real quick to the mouse model thing, because that
Okay.
reminds me. This is another thing that, yeah, if you don't have a certain kind of relationship with the literature, some of this stuff will pass you by. But there's a lot of critiques, maybe in circles like the repeat world, of some types of science, but there are also critiques even within science. So one of them was when I was in graduate school, one of my friends was giving her, I guess, dissertation defense, and somebody that I was working with was like, because she was doing stuff with ketones, and was it autism? It was because how they had the ketogenic diet for
Oh, for like brain stuff.
yeah, for children that had seizures. Uh,
Yeah.
what's the condition? Epilepsy.
Yes.
And then they kind of branched out to other mental, issues, brain issues, that seem to be helped with ketones or ketogenic diet. And they were developing rodent models for this. And they had a rodent model, I think it was a mouse, for something like autism. And it was like, they weren't interested in socializing with the other mice as much as normal. And they had like a few quirks. And I remember somebody saying, like, uh, autism, like, that's such a human thing. Like the way humans socialize. I mean, it's really stretching it to say like, oh, this mouse is autistic. uh, and so there are models out there that people within science are. But it doesn't notice. It doesn't stop the science from happening. It doesn't stop it from being funded. It doesn't stop it from being published. Even if most people working with different mouse models are like, I don't believe this as even a model for studying this question, that doesn't really stop anything. And then the other thing is models like, say, obese mouse models or gut issues or whatever the disease state is, I think they were kind of marketed to science, to research, as we're going to give you this animal model that has an issue that's kind of like this human issue, and then we're going to test treatments on it. So like if this mouse has uncontrolled blood sugar, can't control its blood glucose because we like gave it that thing that like kills the pancreas, made it type 1 diabetic or whatever it is. And then you have this therapy, a drug, a diet, whatever it is. That's reasonable evidence that we can move on to maybe humans, or I don't know, maybe like dogs or sheep or something. It's like closer to humans. But that, I think, so that's already a tenable thing, but a use of models that makes sense. But then when they get into the models where it's like they have a model for, say, gut problems or mice getting fat, and then they study the genesis of that disease in the mice. See, because that's different than trying to look at a treatment when a mouse has been induced or is genetically predisposed in a strain to this condition, and looking at a treatment to look at the cause. Now it's like, okay, so you're taking an animal that you bred or produced a transgenic manipulation to produce this disease, and then study things that make it get that disease like harder or faster. And that's kind of what this is. And so this is like the king of inappropriate use of these models, in my opinion, would be that, looking at the genesis of these conditions rather than treatment. And then also some of these models are just wacky, like for psychological conditions when mice frankly don't have a psyche in the human sense at all. So it's just
Hmm.
an odd thing to do. Okay.
Yeah. Yeah. Totally. So, like related to that, I just pulled up the article that I just um, wrote about the ulcerative colitis models. DSS is dexter and sodium sulfate, by the way. Um, and that's by far the most widely used model of colitis in mice. And again, it's not like it would be incorrect to say it's a model of ulcerative colitis, because it's a model of colonic inflammation that is used in IBD research all the time, most common one. And one of the quotes from one of the review papers that I cited says, this colitis is a simple model of acute chemical injury rather than chronic inflammation, and researchers should not use it as a model for human UC.
Yeah, that's
Like somebody, like a scientist wrote that in a published paper in 2020. And this is by far the most widely used. Like, this is what these people in this sugar paper used. They used a couple of different ones, but the main one was dextran and sodium sulfate induced colitis.
Yeah, definitely. There's this problem cannot be overestimated. That the tools, if scientists get their hands on something that they can that they can buy for their lab, it's not too expensive. It can be used, taught graduate students and maybe even undergrads can be taught with competency to use it or whatever. And it has any justification in the literature at all. Like you can cite something so that a reviewer won't just totally kick your stuff to the curb. It will steer the field more than good ideas will steer the field. So it's their and I had a blog post about this back when I had my blog, and I think that's probably, if I'm going to go back to making like video essays of old blog articles, that's probably should be like top on the list, because I think that's like something that is just an important idea, that the tools, like all the way back to cholesterol stuff, like it's cheap to look at the cholesterol of patients,
right?
So that in and of itself was a huge driving factor. You can just measure it, so it is more important because you can measure it easily.
Right. When you have a hammer, everything looks like a nail.
Yeah. I guess that's a really complicated way to reify that folk wisdom.
Yeah. But, I was actually really surprised in doing research for that blog post that, like, all of these review papers, nobody was hiding the fact that all of these models of ulcerative colitis are not, like, don't have direct translational relevance to humans. Like, it was like common knowledge to them. And yet, in the media headlines and just in the lay population, like, people all over the place will talk about, like, certain additives being, like, horrible for the gut or even causing IBD based on mouse studies. Like, over and over, people do that. Like, there are examples, and I have them in here, of treatments that are now mainstays in the treatment of human IBD that initially weren't successful in mouse studies. We have the other way around, where things that were super promising in mouse studies actually made things worse in humans. And
did they help the mucus in the mouse studies but hurt it in humans, or was it the other way?
Oh, I don't know. Maybe both at the same time.
Yeah.
And, like, there's even I was on the website of this one company that sells, like, mice for mice models of ulcerative colitis, like gene knockout mice or whatever. And they had, like, a table to help you decide, like, which model to buy and that kind of thing. But there was a bottom row of the table that said translational relevance. And for two of the most common models, it just said no evidence on the relevance to human disease. So it's just like, and, like, nobody talks about that.
Well, no evidence isn't evidence of no.
Right. And yet.
Yeah. So it's actually, it's on you to prove that this study doesn't have translational relevance.
Right. Totally.
Because for all we know, so. Yeah. It's funny that these subfields are created, like, they'll get a thing they can do and a few groups will do it. Say it's this mouse ulcerative colitis model. And then they'll just, like, sort of admit on like page 6 or whatever that this has nothing to do with human ulcerative colitis. But then a whole subfield of the literature of like, hey, what stops mouse ulcerative colitis model will like live on and be funded like by, say like NIDDK, like that part of the National Institute, kidney and digestive, the stuff that deals with a lot of the internal medicine, biomedical research. That was what my lab in graduate school was funded by. Because there's just a disconnect between the, and it's just and then the reviewers for the one paper will be like the other people that do the same thing or something, and it's they're just like, oh yeah, this technique that both like all 3 or 4 of our labs use to just keep publishing papers and getting funding and like getting promotions at our like positions. This is great. You can publish that, and they're the expert in it, right? Because they're the only other people that do it. Like nobody's there. Like there's no adult in the room to be like, why is any of this happening? Just, you know what I mean? Just stop.
Yeah. Yeah. And then the media is like, whoa, sugar?
Yeah.
We can run with that.
Yeah.
Sugar causes disease?
Definitely.
All right. Yeah.
Yeah, it definitely does. And, I'd like to point out, you don't happen to have on hand the amount of sugar in these experimental diets, do you?
Oh, so I have the paper open.
Is it more than 35% of the calories?
Let's see if I can find that quickly. What was interesting, though, is that they tested, and actually, the whole reason I opened this paper is because I thought it would be fun and interesting to write a blog post on, like, does sugar cause ulcerative colitis? Like, especially from a kind of more Ray Peat perspective where, like, sugar is not bad.
Mm-hmm.
And then versus the mainstream perspective where sugar is, like, automatically bad and causes everything.
Mm-hmm.
I wanted to actually look into that question and got totally sidetracked on this whole other thing, but I might still do that. But they used a lot of different
Did they use it in the
diets.
water, or it was all in the diets?
I think it was in the water. I'm not sure I'm going to be able to.
Because I think the most convincing studies I've seen are the ones where it's in the water, because then they're just really loading the mice with it. I mean, it's convincing, but it's also ridiculous, because there's really nobody out there saying you should just put sugar in all of your drinking water all of the time. So that's a little bit of a red herring. I mean, I guess you could liken that to drinking soda. But yeah, the reason I mention that is because, like in the previous stream with, or was this on, whose channel was this on? Anyway, I had pointed out that the controlled diet formulation for research diets, just controlled diet, just the stuff they sell to everybody for normal mice, is 35% of the calories is sugar. Which I would call a high sugar diet. And now, of course, this is not being necessarily fed to mice that are predisposed to this disease or have been given this chemical or have a knockout of interleukin 10. But all the same, mice don't seem to be getting ulcerative colitis or mouse ulcerative colitis from research diets, controlled diets. So that in and of itself, and that experiment has been done probably tens of thousands of times.
Right.
So.
Yeah. So this is all saying 10%, which I think, and I didn't make a note of this, it must have been in the drinking water.
There's something in the water.
But the interesting thing is they tested glucose. They also tested sucrose and fructose. And by far, the most cholitogenic effects were observed with glucose. That were not observed with sucrose or fructose.
And did they still call that sugar in the discussion, for example?
Mm, in the discussion, I don't know.
or just in a in the not necessarily the formal discussion, but the part in which it's discussed or described, maybe in the results in the.
I think they usually specify glucose.
So is it the uh, press, popular science press that?
Oh, calling it sugar?
Yeah.
Yeah, yeah,
Okay. Okay.
yeah. Well, actually, they keep going back and forth. Like this one heading of this section says sugar-induced colitis pathogenesis is microbiota dependent. Then the next heading, glucose-induced colitis susceptibility is transmissible.
Yeah. Well, when they put water against glucose, sucrose won.
Right,
Or
yeah,
sugar won. So
exactly.
they've got, yeah,
You got it.
they're
Got it.
using the stop being confused about health model of describing their uh, materials
Yeah,
and methods.
yeah. No, I think it would actually be interesting to talk about um, like this particular study and like the sugar colitis question another time, like after I've like looked into that. Because I remember finding it really interesting that the control diet to keep it like calorie matched, I believe used like extra like cornstarch or just some kind of starch
Hmm.
in the control diet to compensate for the extra sugar calories. So it's like still glucose, right? Because
Yeah.
it's starch.
Right.
So it's like there was some difference between the glucose and starch glucose in these mice for this experiment.
Interesting. It
Yeah,
seems like it would be the palatability, or because that would be
yeah,
sweeter on the tongue. Um,
yeah.
but
I
then
took,
so would sugar, or as in sucrose. So
right. Yeah, we can talk about that another time because I took a lot of notes on this, but then got sidetracked with
yeah,
the
that's the problem.
acromanthine muciniphila
Yeah,
thing.
that's a problem with diet experiments is you can't have controls. Like when I did my um somebody just made a funny comment about stop being confused about health. uh they said I challenge you to a duel. Which I guess I do. But anyway, when I
Yeah, you did that a while ago.
yeah, when I was doing my fatty acid studies, there was no control. So I had a control that was a low fat diet, but that isn't a control because it takes away so there's no way to control. Okay, we have the effect of saturated fat and the effect of polyunsaturated fat, mostly omega-6. They're kind of controls for each other, I guess, because they're the same amount of fat, but you can't have okay, take both of those away, because you're not just taking
Right,
those away, you're also taking away fat. And you're not just taking away fat, obviously you're replacing it, because even if you just take the fat out of that diet, the mice will eat more. And in so doing, they will get more whatever the carbohydrate is in this diet that has been defatted, or the protein in whatever ratios, than the others. So it's not just an effect of removing those fatty acids. It's not even just an effect of removing fat. It's an effect of removing those two things, which are related, and adding something else in compensation, because the mice are just going to do that, to eat
right.
more.
Yeah, yeah. It's not like testing a drug where it's like the drug or the absence
Yes,
of the drug.
exactly. And
Yeah,
that is also something that the average person talking about science doesn't understand. That controls, people just say there's a mantra where they say, "Oh, this was a randomized, controlled." Sometimes they'll throw in double blind.
they're the magic words.
Yes, it's a magic mantra of the current religion, that's popular with the aspirational class now, the church lady class of like science or whatever, of I fucking love science. And they and it's a mantra because they don't know what that means. And they don't, not only do they not, I mean, they might know what it means in one context, but in different types of science, controls are different. And sometimes it's controversial whether that is the right control. And sometimes you just cannot have a proper true control. And with nutrition, that's pretty much the default is that you can't.
Yeah, I think they know what it means in theory, but in practice, it's not that perfect.
Yeah, in theory. Yeah. Yeah. You know, this whole science as a platonic form thing, or as a quasi-platonic form, is really driving me bonkers. Like people are like, oh, well, no, like science is self-correcting and science is this and science is that. It's like, listen, science is that which is produced by scientists when they're being scientists. Okay? In the same way that, you know, there might be some dictionary definition for poetry. But what is actually in our material reality poetry? It's that which is produced by poets when they're writing as poets, like poets qua poets. And that's what science is. Okay, science as this like abstract idea isn't real. It's not a real thing that you can criticize because it's a metaphysical idea. But science produced is like a material thing that you analyze with a material analysis. And you can't defend a material science with an immaterial definition of science. So when I'm attacking science, somebody could say, well, you're really just attacking this scientist who's doing bad. But it's like at a certain point, the entire institution is responsible. And this is what science is because it's what scientists are producing. So,
yeah. I feel like I'm going to regret saying this, but it almost sounds like a Mott and Bailey situation.
That's right. Yeah.
We don't have to talk about it just.
Yeah. Well, I finally got a handle on that, actually.
Yeah, you did. You could talk about it. I can't talk about it.
Yeah. It's the Mott and Bailey, you know, took a long time for me to just really pound it into my head, which is which. And it's sort of like mortar and pestle. I think when I was a kid, I kept being like, okay, but which is the, like, the pestle is like, right? You just like have to like.
I actually don't even know, like, I don't know that. Which one's which?
It's the pestle is like the tool. And the
okay, that makes sense.
mortar is the bowl, yeah.
Okay.
So yeah, this we're talking about the science thing, and before we get too far on the just trashing science in general, the specific trashing that we're doing on this field, is there any other big points on that?
There was one that, so I kept hoping in all of my Google Scholar and PubMed searching to find just one person acknowledging the disparate results in this very specific field of Akkermansia muciniphila and colitis.
Mm-hmm.
Because I am a layperson. I am not like smart enough, trained enough, whatever, to look at these 2 or 3 studies showing that it makes colitis worse, and then these like 5 or 6 studies showing that it makes colitis better, and parse out why that is. Like I can't be like, oh, well, in this study they used this different strain, and like this what they used this percent. Like I can't do that. So I just wanted to find one person, sort of, to sum up the field, at least acknowledge that there's this conflict happening. And there were quite a few review articles on this bacteria, so I kept thinking I was going to find it. And finally, I was reading one that is called Akkermansia muciniphila, key player in metabolic and gastrointestinal disorders. And I was like, man, that sounds really good. Like,
good title, good start.
This was published in 2019. That's pretty recent. They're going to have to like acknowledge this, like, this controversy that's going on. And first of all, like, a lot of these papers are not very well written. Like, I'll just say it. Like, they...
well, they're like, English second language, right?
Yeah. And, like, that's, like, the fact that they can even write in a second language,
mm-hmm.
I cannot do that.
Mm-hmm.
So that's very impressive. But on the other hand, like,
It's a
if
serious
you're,
business.
yeah,
Hmm.
serious business.
Yeah, should be. Yeah.
Yeah. So I'm reading this article and I get to this point and I'm getting so excited that they're actually going to address this. They're saying because of its ability to degrade mucus, it was supposed that A. muciniphila could contribute to intestinal inflammation because low levels of mucin allow the pathogen to access the mucosa. To support this, trials, yada, 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 did this and that. So, was exacerbated by A. muciniphila, suggesting that this was based on its ability to interfere with host mucus formation and production. However, and I was like, whoa, they're going to give the other side of this now. However,
Yeah.
several trials have demonstrated that amucinophila controls fat storage, adipose tissue metabolism, and glucose homeostasis.
Yeah, that's a bit of a, what's it called? Not an anticlimax, but
It was pretty anticlimactic. Non sequitur.
yeah, it's a non sequitur. It's a non sequitur and an anticlimax.
Yeah. So like I can't overemphasize how disappointed I was
Was this the
to finish
German?
that sentence.
Was this the German
This
authors?
The German one was good.
Okay,
No, this was the German people were the ones who finally like
solved the problem.
gave yeah, they solved the problem. They were the ones who replicated the initial experiment, found not the same results, and were like, hey, this is the state of the evidence here. And in my article, I actually highlighted that paper, like if anybody's actually interested in this, which, probably unlikely, but if anybody is, that is the paper to read because they know what they're talking about. But yeah, reading this review article, it was almost like if you had a couple of friends who were dating or something, and like I don't know, the girl came to you and was like, "Hey, like Paul was cheating on me with Rebecca at work,"
Uh-huh.
and then later on you're talking to Paul, and Paul's like, "I know none of my coworkers. Like I don't have any friends at work." And you're kind of thinking about this, and like both these people, you're friends with both of them, so you're like this is kind of weird. Like they're saying contradictory things, but there has to be an explanation, right? And then maybe you're gossiping with somebody who knows both of these people, um, and she's like, "Oh, like did you hear about Paul and Sarah?" And you're like, "Oh my gosh, yeah, like Paul is cheating on Sarah with Rebecca, right?" And she's like, "Yeah," and he's all like, "I don't even know who Rebecca is. Like that's so crazy." But get this, and like you get all ready to have this friend like resolve this conflict and like explain what's going on. And she's like, get this. Paul has started going to Chipotle every Tuesday. Like, I guess he really likes burritos now. And you're just like, ah.
Does he like it a lot? Does he like Chipotle a lot?
Their burritos are thick.
They come out so thick.
Yeah, like that's what I felt like. And it was that experience for weeks trying to parse through this literature of this quite small field where
Mm.
it would be reasonable to assume that these authors are aware of what's going on.
Yeah, that would be reasonable. That is definitely the assumption that people that fucking love science have. Um, and there are fields for which that's true. Uh, I think my field, actually, quino proteins, bacterial quino proteins, people that work on those they probably know all the other groups that work on those things, and they're not going to get caught saying something that is totally contradictory or exposes them as being very ignorant of the field.
Nice.
But when you get into the sexier stuff, which is the problem with fad, one of the problems with fad topics like gut microbiome, the sexiness of it reduces the need to be like high quality. And so, yeah, it's pretty silly. And the gut microbiome field in general is huge, but yeah, you're pointing out this is like a subfield, and it is knowable.
Yeah, like I'm pretty sure I read every single one of the relevant papers in this small subsection of the field. Like I didn't read all of the Akkermansia muciniphila papers. Like I didn't pay attention to like all of the obesity, metabolic syndrome stuff. But as it relates to gut health, and like it became very rapidly apparent that a small handful of papers were being cited over and over and over by all of the people writing about it. Like I started to recognize the author names and years, and they would say like, "Oh, this was like Smith 2010." I'm like, "Oh yeah, that's the paper that showed that there are lower levels of A. muciniphila in people with IBD." Like,
yes, yeah, that's another good one. That's another, because if there's 10 papers or any number of papers and some group decides to cite two of them, like I've actually done that before. Like sometimes you have to make a choice. I mean, you can't just put, if you write a sentence in your introduction, you can't just put 26 references at the end of it, even if that
right.
would maybe be appropriate.
Yeah.
And so through no intentionality at all, through just randomness, there will be papers that somebody decides to cite, and then somebody who reads that paper and gets the information from their introduction or discussion will then cite in their introduction or discussion, and so on and so on. And so it'll be like a little bit of a lottery, and each time it, increases sort of geometrically. And so two papers that are the exact same thing should have the exact same relevance. One could be maybe cited never or once, and the other could be cited like 100 times in the same field, just from that random effect.
Yeah. Yeah. And another interesting thing kind of related to that, as I was trying to learn about this, is I noticed very early on that there were all of these people talking about how Akkermansia muciniphila strengthens the gut barrier. And every single time they would cite the same study. And I went to look at that study, and it was an in vitro experiment, like one single experiment on a cell line. And I was
like a mucus
you guys
producing
are all
cell
just
line,
like,
or
like an epithelial cell, like colon cell line or something like that. Maybe small intestine, probably colon. I think it was a human cell line. But yeah, all these people were just like saying this like it was an established fact, and they're all just citing the same one study.
right? Well, that's how it's established. As
By
of
one person once.
yeah, I mean, do you even read the science? I don't know. Like,
I guess.
somebody's talking about Thomas. Who's Thomas Delore? I'm just like scanning the comments here. I blame Thomas DeLauer. Oh, Matt said that, and then somebody said Thomas DeLauer looks like he needs some sugar so bad. I Google image searched this guy. He doesn't really look like he needs sugar. He's like pretty buff.
Yeah, I don't know who that person is.
He's definitely on anabolic steroids, and he looks like he's some kind of, guy that's probably selling something. Definitely not a natural physique, but yeah, I don't know anything about that guy. There's too many people to know. Like,
Totally.
yeah, it's just, can we cut? can we? This message brought to you by Bill Gates. Can we depopulate the influencer network so that it's more manageable? Bro, somebody said bro scientist beefcake. Yeah. Okay, fine. Yeah, he looks like, I mean, when you look like that, you're going to be bro science. Keto. Okay. Also, somebody asked, there's one question, how does PUFA relate to the microbiome? That's probably a really, really big question. The one thing that I had been writing about that I feel pretty good about, and that I feel like this is the reason why PUFA, dietary and also supplementary can have some acute beneficial effects is that it inhibits, let's say, compared to saturated fatty acids, eating and digesting and absorbing them, it inhibits the absorption of endotoxin. So if you have a fairly high population of endotoxin bacteria, producing bacteria in your small intestine, you will have less postprandial endotoxemia, consuming less saturated fat. Saturated fatty acids make something called lipid rafts that help shuttle the endotoxin across, and for some reason, probably having to do with the double bonds and interaction with water, polyunsaturated fatty acids don't really do this. And they also don't help get the endotoxin to human cells, like immune detecting cells that then cause inflammation. So all down the line, PUFA actually kind of, if you have a situation of postprandial endotoxemia, PUFA in an acute manner inhibits the negative effects of that. And I think that's a big reason why people see any benefits at all from consuming that. But that doesn't really speak to the gut microbiome itself. Like, I don't know. Fatty acids in general, high concentrations of free fatty acids tend to be antimicrobial, and I'm pretty sure saturated fatty acids are more antimicrobial, but for whatever reason, that does not make up for in people that probably have some type of maybe minor issue of like SIBO or gut barrier compromise. That doesn't really make up for it in the postprandial endotoxemia, because it does seem to be a fairly consistent effect. I'm assuming you haven't come across anything.
I haven't really looked into it, but for the lipid raft thing, is that, I mean, is that not just an effect of like, would you not get the same benefit from replacing saturated fat with like carbs and not PUFA? Like, that doesn't sound like a benefit of PUFA. It just sounds like, oh, compare, like saturated fat does this thing, and if you replace it with something else, that won't happen.
Yeah. As far as the gut, yeah, sure. That would happen, but I think,
Okay, like that's not even a benefit of PUFA, right? Or
well, I mean, it's people will generally replace. Like, if you tell people to not eat saturated fat, people may cut down on fat, but generally they'll just replace it. As far as in the system, I think it's more of a benefit, because if you do have kind of PUFA in the system, then they actually do the lipid raft thing in the system as well.
okay.
And that's how they that's how endotoxin tends to get to the TLR4, the toll-like 4 receptor 4 thing that is supposed to be the real big turner oner of chronic inflammation from endotoxin in the system. And for the longest time, people were just saying, "Oh, look, fatty acids activate this receptor." There's all kinds of papers, and then there was just this one paper that was like, "Nah, it's." They do, but they don't. They and I really liked this paper because it really like did something that was interesting, which is it showed that all of the reagents that these people were using in vitro, and I guess sometimes in vivo, to use into mice, but a lot of this was in vitro. At least the reagents are not taken from animals, but produced and sold commercially. They're all contaminated with endotoxin.
I remember you said that.
Right. So
Yeah, that's
when they decontaminated them, or I think they found like one or two brands that just had like undetectable levels of endotoxin, all of a sudden magically the effect of saturated fatty acids to activate TLR4 disappeared. It was a really rock solid case that they made, and they made the case that it was just really good. I liked it a lot. So let's see. Think black cumin seed oil curing everything but death makes up for the PUFA. I don't know anything about black cumin seed oil. Do you know anything about black cumin seed oil?
all I know is we sold a ton of it at Whole Foods when I worked there. Everybody was all about that stuff.
Yeah, you told me that once. I think you said that it was like from your perspective as somebody selling things at Whole Foods, it seems like a really big thing in the health community because
Yeah,
many
it's
people will
very
buy it.
expensive, and many people still bought it.
Well, hey, that's Whole Foods, right?
So that's
That's their tagline.
yeah,
Everything in here
very
is expensive,
expensive,
and you'll
but
still
many
buy it.
people still
Yeah.
buy it.
Somebody asked about the carrot salad. A lot of people mention it increasing bloating / bacterial count. I looked into that a few years ago. Um, all I found was that was true, but either all or most of it was done in rabbits. And they have a different digestive system than us. They're hindgut fermenters. So for carrot fiber to increase or even decrease bacterial counts in such an animal as that is, I would say, kind of tough to make, unless unless it's very established in other gut experimental fields that rabbits are a really good model for humans, which I'm not aware of that, and it doesn't really make sense. I mean, that's why rodents are used because they're not
Yeah,
hindgut fermenters. Um,
yeah, I'm not aware of that. I'm just stuck on the fact, like, just the idea of like doing some experiment where you're feeding carrots to rabbits,
yeah, yeah. It's like
and
do
seeing
rabbits
what.
like carrots? Yeah. Yeah, they should have like on the like science. What's one of those popular websites that writes articles about?
Science Daily.
Yeah, Science Daily. They should have like the picture of like Bugs Bunny with the
Yeah,
kid like, hey, what's up, doc? Like, is this carrot going to increase or decrease my bacterial counts? Hey.
yeah. Well, yeah, and then like the step further of like the question of whether these carrots are like increasing bloating for the rabbits. Like,
Yeah, well, is it making big
did
chungus or
Bugs Bunny
not?
have SIBO?
Yeah, big SIBO chungus. Um, so, yeah, the carrot salad. So my understanding of the carrot salad anyway is not that it's supposed to decrease. I mean, okay, Ray has probably said that one of the things it can adsorb and usher out of the system is bacteria. Uh, but I believe his primary recommendation for it is that it adsorbs estrogens and possibly some other things that are left over in bile secretions that will get taken up again absorptively in the small intestine and not get out. And if somebody has a lot of estrogen in their system and they have a gut that is reabsorbing a lot of those things that are getting secreted out, it can really cut down on their estrogen signaling to usher those things out in between meals. That's my understanding of the primary therapeutic effect. The antibiotic stuff, I think, is kind of secondary. He does also say to add the coconut oil, which I did say the saturated fatty acids are not super effective, but the shorter chain ones are supposedly more effective for being antibiotic. And I'm sure when they're mixed with carrots, peeled long ways
Lengthwise.
to preserve the long
Very important.
fibers, there's a synergistic effect you know, because there is.
Totally. Yeah,
Uh,
I still can't. I tried that once. Like I peeled a carrot long ways. Took a very long time.
yeah.
And then I tried to add coconut oil to it. The carrots are cold. The coconut oil solidifies. It's disgusting. I don't know how people manage it, frankly.
Yeah. I've done it before. Yeah, if you
Microwave your carrots. Does that help the long fibers?
yeah, long ways. If you're right, the coconut oil does kind of solidify, but if you like mix it up really, really fast, it'll distribute evenly enough that, like, if you just splash say a teaspoon or something onto and let it sit for 10 seconds and then try to mix it, it'll kind of be solid. But if you mix it right away, it won't. I mean, this is,
Well, then it's like a film.
yeah. I don't think it's bad. I don't think it tastes bad. I think the carrot salad with a little bit of coconut oil or olive oil or both or whatever, not super cold, maybe left out a little bit to bring close to room temperature, or at least maybe halfway between refrigerator temperature and room temperature, with salt and black pepper, I think is good. Yeah, it takes a super long time to do. It's really annoying. And I personally have not had a benefit from it, but I've never done it for more than 1 week straight. So maybe if I hit 2 weeks straight, I would notice something. I don't know. Maybe someday I'll have some live-in waifu to do it for me, and I'd love to do the experiment then. Until that time, I don't see myself doing it unless somebody invents a carrot processing machine that specifically does the
Oh my God.
yeah, takes a carrot and makes it a repeat carrot salad or at least processes the carrot and then you can add the rest. Then you've got something for me.
Yeah. I really like it with just apple cider vinegar and a little bit of salt.
Oh yeah,
And
vinegar.
just
Sorry,
skip the oil altogether. I'm actually a big fan of that. But yeah, I've been meaning to try to do that more consistently. We should just do that. We should just commit to a 2 week.
One thing that I did, I tried making matchstick. Cutting the carrots and making long, because those are actually, first of all, it's way easier to do. Second of all, they're in like real salads and they're pretty. They look good. The texture is good. It's just way easier to do. Now, the grating of the carrot longwise, it's so funny to say. It has this mystique I do not understand. Somebody just said, I just eat a spoon of coconut oil while chomping a carrot. Yeah, right? Why not? But it's funny, I don't think he's ever mentioned that you could just eat the carrot.
Eat the carrot?
Yeah, because it's long.
You're chewing up the long fiber.
Yeah, you're chewing it up, right?
Anyway.
He does not say to swallow it. So...
Well, he doesn't say to chew it, so maybe you're just supposed to swallow them whole, like spaghetti.
Well, he says eat.
Like how my cat tries to eat my shoelaces, just directly down the throat.
Right, like a cartoon spaghetti consumption.
Yeah. Do you have a food processor? Because, if they have the top thing and, the spinny grating thing, you can put the carrot in the top thing lengthwise, and that would accomplish it.
It's kind of spiralizes it or spaghettifies it. Is that right?
No, I think it just it does the grating action, but just instead of putting the carrot in this way, if you put it in this way, it just grates it.
Oh, yeah. Okay. Do I have a food processor? I think I have a food processor attachment for my immersion blender.
I mean, I question how important the grading direction is, but I would actually like to do a trial of this for a couple of weeks, and I would also like to look into what people say about it and what they're basing it on, and I think that would be fun to do a stream about, just a dedicated carrot salad stream. We can eat carrot salad.
So my problem with that is I would feel like I had to do a literature review. Because
that's what I'm saying that I want to do.
Oh yeah, because I started that process, and
did you?
I years and years ago when I first heard I mean, this is 2015, maybe earlier, and that's when I found the rabbit stuff.
Oh right.
Somebody says boof the carrot, insufflate the carrot, inject the carrot, degree in carrot salad making. In before Abhishek. Old people do la formal meals. You guys into starch? ? I'm into starch. You into starch?
I'm into starch.
Yeah.
Starch does better things for me than fruit.
Yeah. I eat sugar and I eat some fruit. Mostly I guess I would say mostly frozen fruit in smoothies actually, and so that would generally be after a workout. Um, I don't know if I do better on starch. I do know that a very high fruit sugar diet, like one time I was doing like a bag of grapes and like some cheese or whatever for lunch. That wasn't good for
That would
me.
do bad things to me.
Yeah. So like if I just had cheese and like bread, like regular bread, that actually like treats my gut better.
Yeah. Same, except the gluten thing is sketchy for me,
Uh-huh.
but cheese and gluten-free bread would definitely do better things for me than
Yeah.
grapes.
It seems to be like my issue is mixing sugars and pretty much fats or really anything
Mm.
that digests significantly slower than sugars.
Oh man, didn't you know you have to eat fruit by itself? Food combining
Yeah,
101.
food combining is I kind of do fuck with food combining because uh
It I think it has some merit.
it's just the
The thing
sugar
I
thing for
remember
me.
though, like I remember having a food combining printout on my fridge for a really long time, and they were very specific about melons in particular. They're like melon must be eaten alone.
Yeah. I had what did you say you had that from a book?
It was like some kind of printout. I had it on my fridge.
I think that was from a book. I had like
Did you
the
have the same
guy
one?
because when I did the raw vegan thing, I collected all of the books of the people that
Mm,
were the person on that, like the enzyme book, the one that has the apple with like the key. Um, and yeah, this
yeah.
one guy that had the food combining thing, and yeah, he talked. Yeah, melons. Yeah, that was like yeah, big deal, big deal.
Big deal.
Mono
Big deal.
meal only.
Only, yes.
Every time I try to reintroduce a starch, I get an emotional train wreck and bad digestion. Well, I'll tell you
Hmm.
what. There's definitely a cadre of people in the repeat sphere that say things like that. I don't see. The thing is, I don't think there's anything wrong with eating a lot of fruit sugar in any kind of chronic way. The only real problem I have with like the fruitarian diet is just they don't eat anything else. Um, it may be that much fructose for over and over and over. If that's like, I don't know, a really, really, really high majority of your calories, maybe that's not so great. But other than that, that was me for a long time, doing well with rice, potato, and occasionally wheat. Starch makes me depressed even more than I already am.
Man.
That feeling when eat starch. Um, I only do carrots from space personally. Okay.
Ah,
Uh,
I'll take that under
Earth carrots
consideration.
have too much oxygen and may poison me. That's definitely true. Well, if you put your carrots in, you got to get a soda stream and a carbonator and put your carrot. So instead of like soda, you put your carrot in the thing and pump in as if you're carbonating a liquid, but you're just, you know, decarbonati de carrot, and you will simulate space carrot. For the record, I approve of starches, and I'm trying to get a better gut to digest it. Hey, goals. So do you want to wrap up the topic?
Yeah, I don't really have anything more to say about it other than it was just a real experience learning about this bacteria and like parsing through the literature on it and again encountering scientists not knowing what's going on in their own very small field.
Yeah, that experiment you did, I find, is replicable. um, that meta experiment. So,
Yeah.
uh, yeah, that's it's funny. Like, there's the way science, the incentive structure of modern science. You really have to understand it to navigate this stuff because it's like if you went into political science and political analysis naive to people wanting power and money or something, and you would be endlessly confused as to people's behaviors. And, nothing would make sense to you, and you'd constantly be pulling your hair out and everything. And you just have to, Danny Roddy says thanks to both of us.
Oh, thanks, Danny.
Thank you. So yeah, like even in my field, it's funny, like people will, like I'm just thinking about, because you said learning about this bacteria, in my lab, we're trying to find new proteins to study. And so we're looking at these bacteria that have proteins that, like, the sequences are homologous to proteins we've studied before. And some of them actually are fungal proteins. And we were, like, oh, I wonder why the fungus produces this protein. And it's, I think the organism is Postia placenta, and nothing to do with the human placenta, but it degrades, like trees that fall down and stuff, like woods debris, forest debris. And I was like, well, it's because a lot of these proteins that I study are actually antibiotic. They oxidize amino acids and produce hydrogen peroxide. And they're often in bacteria that live in biofilms. So they kind of compete. They're like sending out, and I think they even self-regulate their own population that way, because biofilms, they have to be kept in a range or else they overgrow and kill the film. The film is kind of like it's a limited hangout, if you will. There has to be a certain amount that they produce the kind of mucilaginous stuff, the slimy stuff that you would perceive from touching the biofilm that protects them, but there can't be too many.
So it's not always chill for more to chill there.
Yeah, it's not always chill to chill there. So for this fungus, they were like, oh, I wonder what the effect is. And I was like, it's probably also antibiotic, because, they're breaking down dead organic matter, and they're probably competing with bacteria. And everybody was like, huh. Like that was like this profound thing to just sort of, and you would think, that's just so, so obvious. And you just multiply that times a million and look at the incentives, and especially in these fad things. So in conclusion, probiotics. I would say don't believe the hype. And this is not the kind of thing, barring, there might be one or two products that are sort of pretty, have been shown to have some good therapeutic potential. And then I
feel like probiotics like warrant their own like dedicated stream.
Okay, yeah, but people just care. People are going to want some kind of like the overall perspective, just because it's been sort of quasi discussed.
Yeah.
My overall perspective is there's a few proprietary strains that have a decent amount of clinical science. And then there's the endospore products, which also have their own subfield of clinical science, showing some decent efficacy. That's probably like less than 10% of the probiotic market. So what I'm saying is, 90% of the probiotic market is more or less in the realm of speculative, except it's being marketed in such a way that I think calling it speculative is very generous to the people. And it's really, it's more like with the opportunistic. It's people making a company on sort of something that has reached mainstream. Like mainstream people know it when they hear it. They see it on the shelf. They assume something's on the shelf. They assume we live in a country. They assume we live in a society. And in so assuming, they think it's on the shelf, therefore it has to do something good for me, right? And that's really all the average person thinks. They see it on the shelf somewhere. Well, I had a course of antibiotics 6 months ago. I've had a little bit of digestive issues since then. I'll just, that's actually big normie on this stuff. And big normie is really getting, I think, abused by this field. And then on the research side, it's being abused, like in stuff that we actually talked about, that you actually talked about, very questionable lineage and a string of research for people that are, getting funding to do it and publishing these papers. And there's a really good argument that a lot of this should just kind of go away, and that the gut microbiome field should shrink down to the core of things that are either research relevant or medically relevant, which is, like I said, on both sides, on the commercial side and the research side, a very small amount of this big popular field is producing all of the useful information, and everybody else is just me too. So, you used to be able to say me too without it being about sexual harassment. Tag along researchers, that are just picking this field because it's a big field with full of funding. So what is your takeaway? Don't mess
on
it up.
probiotics, or just of this whole stream?
First probiotics, and then from the perspective of a product, which is how most people are going to interface with it, and then from the gut microbiome research.
Okay. Yeah. I mean, like I said, I think that I have not looked into probiotics and all the different products on the market extensively enough to really have a stance. Like I said, Lucy has done that for soil-based probiotics in a really thorough way. I hope that she does that also for non-soil-based probiotics, because I don't really want to do it, but I would love to have somebody that I trust do it. I think that they can provide benefit to people, but I think I would agree with you that most of the ones on the market, they're not specifying their strains. So because if you read Lactobacillus acidophilus, that is not the strain. The strain has to have letters and numbers after it. If it doesn't have that, and it doesn't have that on their website, then it's probably not going to be they're not guaranteeing any specific effects based on research. They're just kind of throwing some bacteria out there that are trendy. So yeah, I mean, I'm not anti-probiotic, but I do agree that it's overhyped, and that people generally misunderstand how they work, and that there needs to be a lot more scrutiny of the actual products to make sure they do what they're purported to do. And yeah, as far as the field, I would just agree with what you said as far as it's gotten a bit ungainly, especially
it's bloated in the gut
Good word.
area. The gut of
Possibly from carrots.
yeah, it's there's a dysbiosis in the gut microbiome field, and perhaps some hypoxia is warranted.
Well, the hypoxia would be good. We want that.
Yeah, cut down on.
We like hypoxia.
Oh,
There's
yeah.
too much oxygen.
Yeah, we need to take the oxygen out of the room.
Yeah.
What you said about strains, that reminds me. When I did quality control for a probiotic product once before graduate school.
Really?
Yeah, there's a product called Good Belly. It's actually oat based, so they ferment oats and then put them in juices.
Sold them at Whole Foods.
Hell yeah!
I stocked them on the shelf.
You probably stocked cartons that I had watched go get
Wait,
filled.
what years did you work there?
From 2011 to 2010 to 2011.
Nah.
But these
I worked there in 2017.
Oh, that's way. Yeah, I was gonna say they probably could have been sold into 2012 maybe even 2013, but definitely not 2017.
Okay.
But the strain that we used was Lactobacillus plantarum 299V, which is one of the ones that has, it's proprietary. It has a little bit more research behind it. Mm-hmm. And I always thought it was funny because every time I looked at it, it reminded me of the planet in Alien that they go to. It's LV2. It's just, if you just look at the letters and numbers really fast, they kind of have a family resemblance. And yeah, you're right. With if a strain if a strain doesn't look like it's one of the planets, like the planet in Alien, then it's not a strain. It's a species.
Yeah.
That's something to know.
Yeah.
Somebody asked, make a video on endocrine disruptors. That's a big topic. I actually worked in that field. That was the field of my lab, my estrogen lab. It was like estrogen and endocrine disruptors, specifically with estrogen receptor. So I could probably talk about that, but that's another thing that would make me want to do a big literature review. So it'd be a lot of work, but maybe.
Yeah.
Okay. Final thoughts. Oh, hey, I'm going to put all of your stuff in the description, all the stuff that you want me to, but if you want to,
yeah, I'll send it to you.
Orally say your website where all your stuff is.
Okay. My website is alyssaluck.com, which is just my name. So it's easy. And actually I have, I guess, a question for your audience, which is, or two questions. Maybe you can answer the first one, which is are there people like talking about gut health and particularly like digestive diseases from a repeat perspective, like in a like sincere, heavily researched way? Because I haven't actually seen that, and would be like, I'm very curious about all of these things that he says about gut health, like the carrot salad thing, the mushroom thing, like just his general ideas on gut bacteria broadly. And those are the kinds of things that I would be interested in doing some sincere lit reviews on and learning more about. Yeah, I guess I'm just curious, like, A, if there's anybody out there already doing that, and B, if people would be interested in that kind of thing.
Yeah, I think the probably the median answer would be kind of everybody in the repeat thing talks about that a little bit, but nobody talks about that exclusively, or like, 75% or more of their stuff is about that. So I guess the answer is no. And I think the second answer is probably, I mean, people are going to say yes to that because people like content that has quality. And then, okay, so the last thing that I'll answer, what's your next video on? If it doesn't change, next weekend, I'll be streaming, I think, with Leo again about, what's that book?
Pottinger's Cats.
Pottinger's Cats, which I said, we were going to talk about a really long time ago. And then we're also going to talk about some of this science stuff and some of this Instagram science, nutrition influencer, like this beef that I've sort of been half jokingly, half sincerely pursuing. So yeah, that's pretty much it.
Cool.
Thank you everybody for watching. Stream out.